Well, good evening, everyone. Welcome to our Working for Clean and Healthy Lakes webinar series. I'm Andrea Lamoureux, the Vice President of New Hampshire Lakes, and I'm so pleased to have so many of you, about 125 folks, are planning on tuning in this evening for our Cyanobacteria Monitoring in New Hampshire Lakes webinar. And thank you for those of you who introduced yourselves into the chat box. A lot of friendly faces there and a lot of new ones too. So exciting. All right, now I'm gonna move this here and hopefully, well, let's see here. My presentation is there. Let's try this one more time. Screen share. Here we are. All right, see if that works. All right, so cyanobacteria, that's a lovely picture there. Amanda is going to tell you all about that, what that is, and why you might not want to see that in your favorite lake. Just a few housekeeping things before we begin this evening. This session is being recorded. Participants, you will be muted for the duration of the webinar. We just have so many of you. It would be too much of a party for you all to be able to talk at once. If you are welcome to leave your camera on, just remember that others can see you. You're also welcome to not have your camera on. Please do submit your questions in the chat box during the presentation, and my team, Crystal and Jessica, will help to answer those as we can. And questions that they can't answer, we will of course direct to Amanda, our expert, at the end. And after this session, you're going to receive an email asking you to evaluate the session. Let us know how we did, what you liked, how we could have done better, and things you want to learn about in the future. And tomorrow morning, you'll receive an email with links to the recorded session and also links to Amanda's slides. So your host for this evening, again, I'm Andrea Lamoureux. Also in the house here, Crystal Costa Balanoff. She is our Conservation Program Coordinator, and Jessica Sayers, our Conservation Program Assistant. And again, they will be manning the chat box, doing their best to answer your questions. And if you have technical difficulties, they can try to help answer those as well. They truly are the experts there. So New Hampshire Lakes, I know many of you, this is your first introduction to New Hampshire Lakes, and some of you, you've been with us for the long haul. We are the only member-supported nonprofit organization working for all of New Hampshire's lakes. And we are a member-supported organization, and I hope that if you're not a member or not a supporter, you do consider supporting us. You can go to our website and find out all the different ways you can help support our important work, nhlakes.org. Our mission is really to keep New Hampshire's lakes clean and healthy now and in the future, and we work with a variety of partners throughout the state, promoting clean water policies and responsible uses, and hopefully inspiring the public, folks like you and the people you know, to care for our lakes. We have a variety of programs. We advocate for lakes and we run conservation programs. You may know us through our pretty high profile Lake Coast Courtesy Boat Inspection Program. We've been doing that for nearly 20 years all over the state, and we've met 1 million boaters through that program, trying to prevent the spread of invasive species. And we've recently kicked off our Lake Smart Lake Friendly Living program. And we do outreach, all sorts of outreach throughout the state. And this year, we have added our webinar series so we can all continue to learn about how to care for lakes from the comfort and safety of our own homes. So again, this is the kickoff of another series, four sessions coming up in July. And if you haven't registered for the next 3, please do. They're filling up, but we'd love to see you there. So without any further ado, we have Amanda Murby McQuaid tonight to talk with us about cyanobacteria. She is from the New Hampshire Department of Environmental Services and is truly an expert in the field. So I'm going to turn it over to Amanda. Should be able to share her screen. There you are. All right, looks great. Take it away.
Okay, and you can hear me all right?
Yep.
Okay. Well, hi everybody. Thank you for having me here remotely. Is pretty interesting, but I think effective and fun that we can still all get together. Yeah, so I am with New Hampshire DES. We have a sort of a new program that's sort of a spin-off of the beach program where we're going to focus more on cyanobacteria blooms and harmful algal blooms that exist on the coast as well. I think that we're starting to see an increased awareness, but also perhaps an increased occurrence of cyanobacteria and other harmful algal blooms in New Hampshire. And so I am still with the beach program, but we are trying to promote this new program. So stay tuned because New Hampshire DES is updating their website, a lot of content that we have available, perhaps some maps in the future. So this is an exciting time for change. And today I'm going to talk mostly about sort of the background of why we care about cyanobacteria blooms, why it's a problem, you know, what's the big deal about it. But this is my contact here on this front page. I have my email with DES, but that phone number there especially is a very quick way to contact me. If you ever see something that you are just curious about in the water, if it looks like the water turned a different color, please take a photo and send it to me. You can text that photo to me at 848-8094, or you can also email me. We have a new email, HAB, harmful algal bloom, at des.nh.gov. Just a little bit about myself, in case I know a lot of people on here. It's really nice to see familiar names, but I've been with DES for a few years, but I've been in New Hampshire for several years, and I worked and studied at the University of New Hampshire under Dr. Jim Haney and other amazing professors there, but Dr. Jim Haney especially has guided me through a lot of cyanobacteria research, and he's also done quite a bit for the state of New Hampshire. So in this presentation, I'm going to kind of focus on the background about, again, why we care about cyanobacteria blooms in New Hampshire. So, you know, the term cyanobacteria is interesting, but a lot of people call it blue-green algae. It's been known as blue-green algae for, you know, decades, but it's in fact not truly algae. It's a photosynthetic bacteria. So it really has the benefit of being bacteria and utilizing the sun's energy for growth. It's been an inhabitant of the earth for over 3.5 billion years, since, you know, probably the beginning of time. It's in fact been attributed to helping create oxygen on the planet. So this is not, you know, a new thing. It's not an invasive thing. There are thousands of species, and there's hundreds of toxins, which I'll talk about today. They're ubiquitous in the environment, meaning that they're not just found in lakes, but they're found in the air, in the soil, on trees, on lichens. They're a pretty ubiquitous organism. They're also found pretty much everywhere on the planet. So this is a table of a lot of words, but basically this is not a complete list of the cyanotoxins, the toxins that are produced by cyanobacteria, but these are some of the ones that have a lot of research on them. And we know quite a bit about these cyanotoxins that are produced by most of the cyanobacteria. The microcystins are probably the most well-studied. There's a huge variety of microcystins, but we are also focused on microcystins because most of the cyanobacteria that we know produce microcystins one time or another. They're sort of interesting in that they can be genetically regulated, which has a lot to do with water chemistry. So some organisms can produce this toxin, but they can turn it off with their gene regulation. This toxin is attributed to gastroenteritis. It really targets the liver and the digestive system. It can seriously inhibit the protein buildup and can cause tumors over time. So this one over time is chronically something that can cause issues. The nodularins are very similar to microcystins, but they're found mostly in brackish waters. So this is not just a freshwater issue. There's also a very serious one called anatoxin. This is the one that's been attributed to dog deaths. It's very fast acting, and in fact, it just sort of affects the seizures of animals. So it affects the nervous system and causes seizures or death in some cases. There's a whole list of a variety of different types, but in the end, they range from being dermal toxic to neurotoxic to, over time causing some issues with your vital organs. But each type of cyanobacteria is very different. They're like plants in that they have very different requirements for growth. It's not that they all just like hot, nutrient-rich water. There's a lot of other factors that play into why they grow as well as they do. Because they're so different, they have very different sizes, shapes, and they have a very different habitat, in fact. So they do play a different role in the aquatic food web, and they also produce a big variety of toxins, and they can sometimes produce more than one type of toxin. So you might be thinking like, well, I've never heard of anybody getting sick, and there's no reason to really worry about that. It's been around for years. And that might be true, but there are some pretty significant cases that have occurred across the world and over time. And we are seeing more of these effects in recent years. So I won't go through each of these cases, but you could read through briefly here if you'd like. I think the most interesting and sort of devastating one that I had read about was in 1998. There was a hemodialysis center in Brazil that used a contaminated reservoir that had this species called Aphanicapsa, and they injected several patients with contaminated reservoir water, and several people ended up dying from this event. And it was just because they were not aware of what was in the water. Now, Aphanicapsa is actually a very common type of cyanobacteria in some of the cleanest lakes of New Hampshire. So it is something that can be fatal if consumed or injected. In 2018, you might remember in Florida, there was a huge bloom that occurred at both the marine side with the red tide toxin and inland with the freshwater toxin. And what happened was in Florida, they released the freshwater bloom into the marine habitat. And it ended up really being fatal to several marine mammals and caused a lot of fish deaths. It was pretty devastating. But there are a lot of studies that show that when there are blooms that are toxic in both marine and freshwater environments, that they can cause a disorientation and death of several mammals. There was a case in the San Francisco Bay where a freshwater source was leaked out into the bay and killed several otters. There's also evidence for a bioaccumulation of toxins into fish, into shellfish, just like red tide would. Studies with crops, cattle, the list goes on. There's a lot of very interesting studies that have been done over the years and across the world. But one thing that we are looking at in New Hampshire with the University of New Hampshire, in fact, is looking at how these things might become aerosolized. It's just another mode of being affected by cyanobacteria. And there's also been a few studies that show that perhaps cyanobacteria toxins may be related to neurodegenerative disease, such as ALS. There's still a lot of research that needs to be conducted on cyanobacteria, but these are just a few examples to show that it's not just your contact while you're swimming. There are a lot of reasons that we should be studying cyanobacteria and their toxins. I think the most dear to heart for New Hampshire, since we don't really have severe issues with cyanobacteria in New Hampshire, there are a few lakes that experience blooms for several years or have been known to have blooms. But our biggest issue is that we think that we could be protecting our children and our dogs from consuming toxic cyanobacteria, even if there is just a tiny little patch at the beach. So with New Hampshire DES, we're really trying to increase the awareness of this and increase the reporting of blooms so that people become more aware of the dangers associated with toxic cyanobacteria. Because the dog deaths is something that I think is just a little more direct to our understanding of what it can really affect in our lives. There was a pretty nice review study done that looked at the confirmed cases of dog deaths after swimming in freshwater or marine waters and attributed these to cyanobacteria blooms. And the toxicity really ranged from, upset stomach to death, and the illness ranged from less than a day to 6 weeks. So some of these toxins are fast acting and can cause the seizures, the death. Some of them are maybe more mild, but more chronic and can last several weeks and really mess up the gastroenteritis type of symptoms that you might experience if you consume too much of this. And dogs, especially if they go in, you should really rinse them off after they go in a bloom if you notice that they look like they've been in scuzzy water, because then they groom themselves and they actually get an even higher dose of toxin as they are cleaning themselves after swimming in this type of water. So we sometimes also experience what we call fish kills. Fish kills are very natural. There's been several in New Hampshire already this spring and summer. They're not always related to cyanobacteria blooms, but they can sometimes be related. There's evidence that some fish are affected by the toxins produced by cyanobacteria, but most often what cyanobacteria is doing, they're producing oxygen, but then they're taking away oxygen. And it's the oxygen changes in the lake that often attribute to these fish deaths that we see. So just something to consider, that it is natural, but it can also really disrupt an ecosystem. And these blooms, if you've ever reported one, are constantly changing. So you might call us and say, I have a bloom. And then I say, okay, I'll be there in an hour. And then I might get there and they're just like, well, it's gone. And that's just very typical of New Hampshire lakes. And that's a good thing. But it's not truly gone. It might have just sort of blown down to your neighbor's shoreline and they don't know about it. So we will often, try to alert the community when there is a sighting. But these are really truly a timeline of events, so that they can, be blooming and they're up at the surface during the day and then you don't see them at night. They all have a very different daily life cycle, but you can almost see like the ribbons along the shore that really represent a timeline of events that have happened throughout the day as that shoreline is getting more and more cyanobacteria blown onto the shoreline, the beach, your boat launch, whatever it might be. It's constantly changing because the lakes are very dynamic. They can also start to appear in different colors. They're not always blue-green. Once they do get caught at the surface tension, they start to degrade and die. They don't really like to be caught in the surface tension. They start to get bleached out by the sun, and then you might start to see sort of a white streak associated with blooms. And that's a good sign that it is dying. It's decaying, it's bleaching out, but it doesn't mean that you should be saying, okay, let's go swim. It's still pretty toxic at this time as it's dying. It can also kind of look like a detergent, which is kind of odd, but it can really look in a variety of different colors. That really depends on what else is in the water and how degraded the bloom might have become in that time. Often in June, we start to hear from you guys about blooms. And is it pollen? Is it a bloom? Well, we're starting to see that a lot of the blooms actually co-occur with pine pollen. So if you see like a yellow powdery sort of residue on the surface of the water, that is pine pollen. But if you start to see like a green hue underneath it, or if it looks kind of orange, or if it's starting to look brown, you might have a mixture of cyanobacteria, algae, phytoplankton, other things that are sort of creating a lot of different colors. They're not always just a bright blue, green, brilliant color. Blooms can really vary, and here are some examples of blooms we've seen over the years. None of these are recent photos, but on the record, we've just seen a lot of different things. Like these all look like scum, right? We call this blue-green scum, but they don't always look the same. And that's because they're made up of different organisms, and they all have different colors and shapes and sizes, and they are not alone. There's other things there mixed with them. There are also some very unique cyanobacteria that we don't see all the time. Some of them are well known to certain lakes, but they don't always create that blue-green or green scum that we're always talking about. Gliotrichia, for example, is a very common cyanobacterium that we see in two of our biggest, cleanest, most oligotrophic lakes in New Hampshire. Winnipesaukee and Sunapee often experience gliotrichia. We also have this type called nostoc, and they do not create surface blooms. They look like blue-green, green marbles at the bottom of the lake. So you might see a cluster of them. There's other organisms that look like this, but sometimes you might be seeing nostoc. And within those balls are these tiny kink cells to the right there. Those are all packaging toxins, and some pretty serious toxins. So these types you might not see as a surface bloom, but you know, it might be enticing for a child to go and play with some of these. They look like, again, like marbles. And in some countries, they are consumed as a delicacy. There's also types that like to attach to rocks in the sediment. Some that we saw, again, at Lake Winnipesaukee was from the family of the Stygonematales. They really look black under the water and on the rocks. They're very slippery. We don't know much about how toxic they might be or how well exposed you would be, because they are really growing long-term and really covering rocks and sediment. We don't usually see them come to the surface. And perhaps my favorite of the cyanobacteria are the Pico Cyanobacteria, and these are essentially invisible. They are the most numerous organism on the planet. They're in marine and freshwater environments, and we're also learning that they're in the air. You really can't see them all that well until you shine a light on them, and then they shine back. And so these are a really interesting group of the Cyanobacterium that in my recent studies at UNH found them to be toxic, and actually big implications for them to be taken up through the food web, which is a whole other conversation that if anyone ever wants to talk about. I did a dissertation on cyanotoxins and food webs, so I'm ready to talk about it. Not today. Other than to know that the Pico Cyanobacteria are readily taken up through the food web in several directions, and they are also aerosolized. This bottom photo, it's hard to see, it's a little grainy, but we had created these aerosol collectors to see what was coming off of the lakes, and we found that these really tiny ones are the ones that really readily come off of the lake day and night. But perhaps the most common one, I think every bloom I've seen this summer so far, it's very common in June, is the Anabaena. They've actually changed the name. It's now Dolichospermum or Dolichospermum. We see these really tightly coiled, like beaded sort of organisms in the water. And they are, again, really, really dominant in June. We often also see them all the way into November, and that bottom right photo there is a photo from November. But the top right photo is a microscopic photo of what that looked like. And towards the fall, what we start to see is that these start to unravel, and then they release these seeds into the lake. So they're almost always going, they're always ready for the next season. That little cluster in the middle are their growth seeds for next year. So they can really persist year round. And this Anabaena, this is specifically a Dolichospermum lemmermanii, sometimes has this little animal attached to them. They're vorticella. And they have like this really interesting.
You seem to be having a slight technical. Oh, there she is. We
Oh,
lost you for a second.
Should I go back? Am I behind?
We probably lost you for about 15 seconds.
Oh, okay. So I'm still talking about Anabaena. Okay, so Anabaena, or now known as Dolichospermum lemmermanii, is very common, like I was saying, if you didn't hear me before. It is commonly found in June mixed with the pine pollen. But they also tend to have this little animal attached to the colony. These are called vorticella, and they have this really interesting mutual relationship that helps them move around the lake. So these blooms can really look like pollen or powdery yellow, but they also might have this type of cyanobacteria that, again, is probably the most common type that we see in New Hampshire. And these produce the anatoxin that I was discussing earlier. And anatoxin is fast acting and kills dogs. But on a lighter note, I've been getting a lot of other bloom complaints that are not cyanobacteria. And this is a good sign in certain ways, although we are seeing a lot more of what we call green filamentous algae. And people really get nervous when they see green filamentous algae, but there's really nothing to be worried about other than it is a bit unsightly, like blooms are. It's slimy, it's kind of like a ball of like a tumbleweed of lake stuff, and it's very slimy and kind of gross, but it's harmless. It's actually very good to harvest and compost if you find it. It sometimes appears sort of like just under the surface of the water, kind of bubbling, but it's a bright yellow green. It's like a very bright green that's a little bit different than cyanobacteria. And you can also, we have a fact sheet through New Hampshire DES with more details, but you can almost see it like cotton candy, like underneath the surface of the water, where cyanobacteria would come all the way up and really create a surface scum on the water tension. So also know that green filamentous algae is something that we'd like you to report to us, but not to be concerned. On that fact sheet, we talk about something called the stick test. So what you can do is grab a stick, and if you can lift that bundle up and it kind of all sticks together and is slimy, it's probably green filamentous algae. But if you try to poke a cyanobacteria bloom with a stick, it's most likely going to cloud the water and disperse, kind of look clumpy maybe. Might look a little bit stringy like it does in this photo, but you would not be able to like lift it up, and it wouldn't be like a bundle. It would be more of like a striation cloud, and probably not as yellow-green. So, you know, every lake is quite unique. This is in Massachusetts. Both sides. New Hampshire's on the right and Massachusetts on the left. No, it's just something that's very interesting, that every watershed, every lake is very unique. It has a lot to do with the chemistry, but also the biology and the surrounding watershed. So I like this photo. This is borrowed from EPA. They do sometimes have like a little leakage from one side to the other, but for some reason this lake doesn't really get the green color that the other one does. And there are very unique differences between the lakes. So I know everyone's always wondering, so how do we fix this? Yeah, you told us there's a bloom. How do we get rid of it? Well, that's a good question. There's a lot of research out there. There are a lot of treatments, remedies, if you will, that people are proposing that they try. This is a short list from a paper that we were reviewing recently on different treatments that people are using across the world. It's a lot of chemicals. It's a lot of biomanipulation. There's a lot of different techniques. But because each lake is very unique, you really have to understand the lake before you just throw something at it to try to get a quick fix. There really are no quick fixes with cyanobacteria, and the applications of these must be really carefully considered. They can be very expensive for one, and not necessarily going to fix the problem. You might be stuck doing something continuous to really get an effect. It can change from year to year based on what else is going on in the lake that year, what the weather is doing. So there's a lot of unknowns when you are applying certain treatments. There's also several short and long-term effects. So some of these short-term effects might look good, but they might actually create bigger problems for the lake down the road. And we're always wondering, are we creating a more toxic environment by applying any of these treatments? So while New Hampshire is, we're ready to help lakes when we have huge issues, we don't see that there are too many lakes experiencing these issues where we would go to the extreme of trying to treat it with another chemical that's also toxic, for example. So, but we are considering how we could help down the road in permitting certain treatments. What New Hampshire is doing about blooms is, maybe you could consider it as it's an ongoing evolution of what the science is telling us, what is available, what's regulated, what is important. But what I'm seeing in New Hampshire is an increased awareness and interest in the subject. And that might be because we're talking about it more, but I think that people who live on lakes are starting to see changes. They're seeing changes in the watershed, in the development, and just the biota sometimes. You might be starting to see blooms that you never saw 20 or 30 years ago. In New Hampshire, we're doing a lot of monitoring, outreach, research, discussions that span from the locals that live there to volunteers with certain groups, universities. The EPA has done some stuff, and here at New Hampshire DES, what we're offering is a free service for analysis, so we can really figure out what's going on as it's happening. And we do identification, cell count, toxicity, and then we issue advisories. But we also work with certain lake groups that are still wondering what they can do. The New Hampshire DES Watershed Management Bureau in general has a really great system with the Watershed Assistance Program. We are guiding people to create watershed-based management plans. There is some funding that is sometimes available. There's big projects that come out of this, and certainly if you have a cyanobacteria bloom, it often sparks this watershed assistance pathway. We also really advocate for soak up the rain and the best practices at the water's edge to really reduce runoff. And now we are also working with the Drinking Water and Groundwater Bureau, and they have offered grants for public water systems. Up to $10,000, I believe. That might change, but they are here to try to guide people to start monitoring if you are a public water system. And, that might look like, a volunteer lakes assessment group sort of moving to a drinking water system. So they're really open to trying to help anybody who's on a public water system to monitor. But the biggest thing is really that we are relying on you to report your sightings. We just can't get there. We can't see it as it's happening. You guys are there, you see these changes. So, photos are critical and reporting your sightings is critical. Because blooms, they occur on the beach. They might be there when we're at the beach, but they're not always there. They're everywhere. They're at the boat launch. They're at your house. They're in inaccessible locations. So there's really no place on the lake that they're not going to be. It's just when is the wind blowing and where are they going to end up, really? Because once they get caught at the surface, they're just kind of blowing around the lake. So you might think, okay, it's gone. It's no longer at my beach. But again, remember that it might just be on the other side of the lake today because the wind shifted. We usually try to give it a few days to dissipate before really declaring that it's gone. So photos are critical. Please call us 848-8094. I'll say that a million times. It's really a very quick way to get in touch with me. Send me a photo and I can just quickly tell you what we're gonna do, what it is. Let's, move forward. It's also good to maybe note the weather, the wind direction, just for your own notes, but also for us so that we can kind of assess, like, is this the prevailing wind? Is this always on this end? Is this something that we're going to expect every June? We start to see patterns when people start reporting them. While we really do not encourage you to sample, we do say, please avoid blooms. We do have a lot of you volunteering to help, and it has been very, very critical with your help. So again, we ask you to be cautious if you are handling blooms. Wear gloves. You could even wear a mask, which is pretty normal these days to wear a mask and a glove. So, we just ask that you just write a few notes down so that we can contact you and we know where the sample was taken from. What we will do again at New Hampshire DES is that we're going to identify what it is. We're going to enumerate it. We're going to understand how concentrated it is. We're also going to run a few cyanotoxin tests. And if there is a concentration above 70,000 cells per milliliter, that's our state threshold, we will post an advisory. The towns are notified. We ask that signs are posted. We do have a press release statement, we have a map, and we have social media. A lot of these things are going to be changing in the next few years, but our message is just to inform your neighbors, let people know so that they don't just walk into something that could be toxic. And we are working on sort of a bloom response protocol for recreational and drinking water purposes. So what you might see is a variety of different types of blooms out there. If there is an advisory, please check our map. You might also see a sign posted at maybe the boat launch or the beach. We are also changing the sign just to be more visual and also just so you can read it a little bit better. But the message will be the same in that this is an advisory. We're not closing the lake. We're not closing the beach. We just want people to be aware that if you see this, you should really avoid contact and you should really not let your dogs or children swim in it. On our current website, which again will look a lot different in the next year, but if you go to the programs page on the home page, des.nh.gov, you can find under programs the beach program. But the best place to go really is under alerts. You'll see we have a lot of different things that we're monitoring: air, beach, cyano, drinking water. But if you click on the beach advisory, you will get the beach map that will give you the beach bacteria advisories and cyanobacteria bloom advisories. What we also do though is we create a write-up that gives all of the boring details of what the cyanobacteria were. And that's just a good record for us to have because it really lists all the details of what it was. And that can also be found under alerts. So you'll first go to the beach map. You'll see the map looks very different this summer because we are not sampling freshwater beaches. We will be doing a few for E. coli, but we are very short-staffed right now for monitoring fresh beaches and we're struggling. What you'll see though is a legend that will show which beaches are open and which beaches are under advisory. Now, whether a beach is truly closed is up to the beach manager, but we will have a caution icon up on the map if there is a cyanobacteria advisory. There will also be below that map sort of a description of what the warning was. And that press release statement, again, gives the boring details of what was in that bloom, just for the record. That can all be found under cyanobacteria and alerts. We are also using Twitter. We have Facebook. We're trying to expand our social media reach, but photos are the best thing to share. So if you have your own Facebook lake group, please share what you're seeing. This just really helps people become familiar with what it is and knows what to expect if they do live downwind from where you are and it's coming. So another sort of group that we're working with is the EPA, and you know the state does work with the EPA quite a bit, but they have launched a cyanobacteria monitoring collaborative, which we do support. If you go to cyanos.org, you will find that they are really showing 3 different types of monitoring that citizen scientists can get involved with. One is called Plume Watch. That is really an app that we're getting a new app, so stay tuned for this, but the Plume Watch app really allows you to share your photos quickly and should also be contacting me with that. So it's just a nice way to submit your photos, and they do ask for 3 different photos, like one that shows up close of the bloom, one that's more of a landscape view, just kind of showing the extent of the bloom. We also have trialed cyano monitoring, and some of the lake groups might be familiar with this because we did do several years of trialing cyano monitoring where we used a fluorometer to try to determine cyanobacteria in lakes. And then the third component is called Cyanoscope. And this is really for that like dedicated person who is interested in having a microscope and looking to see what they can find under the microscope. So I was going to show you the Plume Watch app video, but it's a little bit long, and maybe I'll wait to the end after questions if we have time. But if you do, go to YouTube and look for the EPA Plume Watch app, you'll get a nice little like tutorial on what is coming soon with this app. Basically, everything that gets submitted through the Plume Watch app is then housed on a citsci.org website where it's mapped. They're also looking, the EPA is looking to advance this mapping technique and the use of the data. The Cyanoscope side of it is an online key that helps you identify what you might be seeing. We have like about 12 really common ones in New Hampshire. So once you start seeing a bloom in your lake, you're almost guaranteed to see that same type of cyanobacteria the next year at the same time. But it can also change throughout the summer. We often see in June Anabaena blooms, but by the end of the summer, we might have a larger variety of different types that pop up all the way, after fall turnover. And this actually worked out quite well with the group in Wolfboro, who were able to use the Cyanoscope and identify gliotrichia and made us aware of it. So I really think that public awareness and the citizen science on this is on the rise, and it's really useful because you're there and you see it as it's happening. So again, your involvement really does make a difference. To be on the lookout for blooms really helps report these events so that we know when these events happen across the state. It also helps to just inform your neighbors. You could actually be preventing someone from getting really sick or their pet from getting sick. We really rely on eyes on the water through weed watchers, VLAP, the lake slay monitoring, you know, all these different groups that exist that are on the lakes, NH lakes. We really appreciate having eyes on the water and reporting what you see. But there are some things that you can do if you live on the lake, and you've probably heard this before, but there are best management practices at the water's edge that really do help prevent stormwater runoff, nutrients, and excess phosphorus and nitrogen that might be getting into the lake that really help feed cyanobacteria blooms. And so with that, I say thank you guys for listening. I know it's not a very exciting topic, but I find it to be interesting. And I think that in New Hampshire, we don't really have too many problems. We just are trying to keep track of these little things that pop up so that people just know about it and go to stay away from it like you would stay away from anything poisonous. So thank you, and I'll take any questions. Thanks, guys.
Yeah, thank you, Amanda. That was awesome. I'm going to turn it over to Crystal and Jessica for all the great questions.
All right. I'm not sure if anybody else is having audio problems, but I'm getting some feedback, so I'll try to power through it here.
You're better now. You're good.
Better now? Okay. So we had a lot of great questions in the chat box. Nice job, Jessica, keeping up with a lot of those. You're so speedy. So I will just go through them one by one. One question, Amanda, was which organisms cause red tide, and how common is an occurrence of red tide in New Hampshire's coastal waters?
Sure. So we have a shellfish program that does monitor for red tide organisms, and we're looking for mostly non-cyanobacteria. They're dinoflagellates mostly. We are starting to see an emergence of species that we haven't really seen in recent years in numbers that have been quite high. I really shouldn't try to quote them on this, but I haven't been directly monitoring for the red tide, but I do know that the dinoflagellates have been increasing in recent years out at the coast. And the shellfish program is obviously monitoring because it has great effect on our shellfish community.
Great. Well, not great that it's increasing, but great information. Another question was about filamentous green algae. They described it as that green cotton candy looking stuff on the bottom of the lake, and it was aptly timed because it was basically your next slide. So you kind of already addressed that already, but there were some additional questions that filtered in about filamentous green algae and whether or not it's a precursor to anything. And I recall having conversations with you about how filamentous green algae isn't necessarily an indicator of excess nutrients in a water body. But do you think that if people are starting to see excessive filamentous green algae blooms, that it's an indicator for a harmful algal bloom maybe coming down the road in the future?
No, I don't think that they're necessarily related. We do have lakes where they're both occurring, but we actually, we're finding that the green filamentous are mostly a species called Spirogyra, Eujotia, Zygnema. These types have been attributed to clean water. They like low nutrients. But one thing that I think we're seeing with Volunteer Lakes Assessment Program, maybe UNH, and with Cyano now is a lot of our cleanest, most oligotrophic lakes are probably most vulnerable to experiencing a bloom with just the slightest changes in water quality. It could be excess runoff, rain. It could just be a really big wet weather sort of event. It has a lot to do with the flow of the lake, the depth of the lake. So again, each lake is quite unique, but we don't know why we're seeing more of these bundles. I have a feeling it has to do more with extreme weather and warm temperatures. But if you read the literature on these species specifically, they tend to like low nutrient systems. They are probably more likely to be competing with cyanobacteria for light and nutrients as well. But they're again, they're like a tumbleweed. They're not attached to anything, and they just kind of free float through the lake, and they're catching everything. So they kind of grow to these sizes that are, you know, again, like slimy and gross. We do have some ideas about purchasing nets that might allow you to easily remove that type of stuff and compost it before it gets out of hand. So that's something we are working on to see if maybe we can just stay on top of it so it doesn't look so nasty and gross and slimy, but it is harmless, does not produce toxins, and they don't seem to be co-occurring in the same lakes, but they might be increasing because of warmer waters, warmer temperatures, and even just the slightest sort of effect on the lake. It could be added salt, it could be stormwater nutrients. There's a lot of things that are considered stressors that help sort of release those filaments, and then they just start to grow more and more.
Very interesting. So, you know, we have this intersection of land use change and changing weather patterns, and also lake use change now, where there are more people out on our lakes, and we have new fun toys and equipment that are out there, you know, sloshing things up, and, you know, just kind of changing the natural interaction in our lakes. So, you know, to that extent, how much can we look to excess nutrients like phosphorus and nitrogen, and even, you know, talking about septic systems and people's outdated on-site wastewater systems, how much can we look to those aspects of algal blooms and, you know, think that there's some kind of correlation?
Sure. I think, you know, there's several reasons to update your septic. Yeah. It's, you know, the E. coli alone that might come out from a faulty septic system is dangerous enough, but it will bring in excess phosphorus and nitrogen and the micronutrients that cyanobacteria love. So keeping up with the watershed, whether it be septic or other sorts of sources of runoff, is really important. You might think, okay, well, this has been 30 years, 50 years, 100 years of this activity. It's not going to change, but really stopping, you know, the external obvious sources of nutrients and bacteria can really help a system overcome the situation. It might require further remediation after stopping the, you know, external sources. You might find that there are still internal issues that are feeding cyanobacteria. But each lake is unique. You have to look at the food web dynamics, the sediment, every aspect of the lake is really helping this stuff thrive. And they easily outcompete other algae. So it's something to stay ahead of rather than try to treat later.
So being as lake friendly as we can be now. I like that. Okay, well, a couple of other really great questions. One was about were they picoplankton? The things that light up. So cool. That was the first time I had heard of that. Very, very cool. So the question was, can these organisms, you know, form colonies to an extent in bloom and cause toxicity concerns, you know, just from being a bloom, or is it largely the bioaccumulation through the food chain that is the concern?
Yeah, that's a really good question. So we don't know a lot about the Pico Cyanobacteria because we need to do some DNA analysis to really understand what we're looking at. But the Aphanocapsa, the type that was attributed to the Brazil deaths, Aphanicapsa is actually considered a Pico Cyanobacterium because the size of the cells are so tiny, but they do create colonies. Like this one here actually is called Aphanicapsa, and it creates these micro colonies, but you can see there's several thousands of cells around it and that it just kind of exists in the water. In my research, I found that this type did produce microcystin. We did not go too far on toxicity. Again, hundreds of toxins out there, but it did produce microcystin. And it's interesting because what we're not talking about are the zooplankton here, and there are animals in the lake that directly feed on algae and cyanobacteria. And they all have their own unique way of feeding. You know, some are carnivores, some are herbivores. So how those get transferred up the food web is something that I am really interested in learning more about. In my own research, I found that there are certain zooplankton that do magnify microcystins and pass those up through the food web. And we think that part of that could be coming from the picos because they are readily consumed. Even at low toxicity, they're just consumed in such great numbers that it adds up. We don't know what other toxins they can produce, but also at UNH, this was the organism that we think people are breathing in. This is the one that's in the air. It's, you know, it's found in outer space. It's everywhere. We might always be exposed to some background level of this stuff. It was also found in lung tissue with another study. So it's a very interesting, very small little organism that can easily become airborne. And I think what we're finding is the smaller they are, the further they will go.
Fascinating. Absolutely fascinating. Okay. Another question was about, you know, just basic statistics in New Hampshire, you know, over the past 5 years or historically, you know, how many cases of illness in humans or dogs have been reported? And, you know, is there like a running log of that, or is it just sort of through the grapevine and speculation?
It's both. It is both. We do have an illness report form. What ends up happening is that doctors, vets, you know, people that are professionals on this are unsure of the connection. It seems very likely, but there's not a lot of good tests, especially, you know, in the past, there weren't a lot of tests to really say, was it or was it not? Some of the veterinarians are very certain that dogs have become ill or died from cyanobacteria exposure. They've done certain enzyme testing on blood, or they've induced vomiting. We don't have a very good record of that at New Hampshire DES, though. Oftentimes these cases go sort of unsolved. It's sort of the same for anything related to gastroenteritis. Gastroenteritis is probably one of the most common reasons that people go to the hospital. And one of the most common answers is, well, it was something you ate or drank or exposed to somehow. And it leaves you like, what did I do? What did I do? What did I eat? Where was I? What did I drink? So I think that there's a lot of improvement that needs to be had or conducted or figured out really on really tracing the two together, really trying to correlate them better. All
right. I'm going to ask one more question, and then I know Jessica's been busy in the chat box, so then I'm going to turn it over to her. Another individual asked if DES is working with the Dartmouth College Cottingham lab on cyanobacteria. I hope I'm saying that right.
Yeah, Kathy Cottingham, she's a professor at Dartmouth. She does have lots of projects going on with cyanobacteria. She, as far as I know, she's doing a lot with gliotrichia on our clean lakes there. She might be focusing quite a bit on centipede in her research, because that organism just grows very differently than the other cyanobacteria. So from what I know of her research, it's a lot of gliotrichia research, but she has a really great depth of knowledge on the topic of toxic cyanobacteria, has had several students doing research projects on this. We don't usually work directly together on blooms. At DES, we're not really doing a whole lot of like research. You know, we're trying to be more of a service for the public and be an outreach for the topic. But I do see in the future, especially with our new program, a lot of collaborations that will come out of this, so that we can really sort of couple, you know, the lakes data, the rivers data, and just have more in-depth knowledge of what's going on behind this event. And in fact, we also have somebody at New Hampshire DES, Kirsten Nelson. She's doing lake trophic surveys all the time, but she's also taking up some really special studies looking at lakes that do have habitual cyanobacteria and trying to get more out of the data so that we can really focus in more on these issues. So I do see a future of collaboration more. And we do work with UNH quite a bit too.
Great. Jessica, on to you.
Sure. Hi, Amanda. We had a question about seeing these blooms in rivers as well. You know, is it a similar situation, or is it a whole different body of, you know, occurrences?
Yeah, it seems to be pretty rare in New Hampshire, but it does happen. In fact, there was a report today on the, oh geez, what river was it? Sometimes, yes, we do get reports on rivers. There tends to be a benthic type that grows on rocks and on sediment that's very slow growing. It sometimes gets dislodged and you start to see these like big blue-green clumps that come up to the surface. That's common in lakes, but I've been seeing it in rivers, and it makes sense because it attaches to a rock. It's not really bothered by, you know, the flow of the river. And then at a certain time, oxygen, temperature, you know, natural changes help it dislodge. And you just see these big blue-green chunks kind of floating by, but they don't seem to persist, at least in any New Hampshire rivers that we have seen.
Gotcha. And I know we could probably spend a whole nother hour on this, but maybe could we touch on, you know, are the nutrients like phosphorus and nitrogen the major triggers for these blooms, you know, majority of the time?
Yes, I think, you know, globally, our understanding is that warmer temperatures and increased nutrients, primarily phosphorus, but also nitrogen, are the driving factors. If you really dig into the research, you find that each species has its own requirement. So it gets pretty tricky. There's a specific nitrogen to phosphorus ratio that some of these organisms really prefer. And then many of these can actually convert their own nitrogen requirements, so they can take atmospheric nitrogen and convert it to whatever they need. So they really have a great advantage over other organisms. And it just helps them really grow very quickly in a short period of time. But other, you know, micronutrients, sometimes iron has been a cause for toxicity production. There's a lot of different sort of, you know, just like plants, like this one needs a little more nitrogen, this one needs a little less. Those types of things apply to cyanobacteria too.
Gotcha. And last question that I have on my end, are cyanobacteria sensitive to pH in the water at all? Or does that not play a factor?
Yeah, it's interesting. Sometimes we think that, you know, maybe more acidic lakes aren't a good environment for cyanobacteria, but we know that cyanobacteria actually increase the pH. So, you know, if your water has a pH of like 8, you might be experiencing cyanobacteria effects and not the other way around. So while they might not really care for very acidic lakes, it's not unheard of that they will grow just fine, but they do sometimes cause a slight increase in pH in the water.
Gotcha.
And some other Well, just some other parameters that you might, if you are looking at, just simple water quality, is that, the Secchi disk will be lowered. Your turbidity is pretty obvious. Chlorophyll would go up, turbidity would go up, and pH would go up. So those are some of the, short indicators that there's something growing and something going on.
Awesome. Thank you.
You're welcome.
Great. Crystal, any more questions on your end?
Yeah, can I squeeze in one question?
We'll squeeze in one more. I know it's a little after 8 o'clock.
Yeah, Amanda, you're being very patient with all of these questions. Thank you.
I love it.
Aquatic invasive species. We know that we have issues with aquatic invasive plants in about 87 water, not 87 totally just plants, but we have about 87 water bodies in New Hampshire that have one or more aquatic invasive species in them. And now we have a handful that have aquatic invasive animals. To your knowledge, we hear some things about correlations between aquatic invasive species and cyanobacteria blooms. What do you know about that relationship? And is there more science developing on it?
There should be more science developed. I am very interested in that conversation because I really don't know. I think I have, and I've talked about this topic with colleagues and peers, of course. There is sort of like this delicate balance between having your healthy macrophytes. The natives are important. These native plants are important to keep, even if you want a beach, because they're taking up nutrients and they are shading out light for cyanobacteria growth. So there is sort of a competition between the two. But of course, we also have to keep up with invasive weeds. We don't want our lakes turning into land. It's something that we really have to carefully navigate. And I'd be very interested to find more research on this topic, because sometimes when there is, a massive death of plants and pulling up of weeds and all of that, you're kind of disturbing nutrients in the sediment that could, possibly spike a little increase in the water column, potentially allowing things to grow really rapidly. And when you're also, removing that shade, and they like the sun sometimes, then you're also kind of just opening it up for them to move in and outcompete the other algae. So I would like to learn more about that topic.
Cool.
Well,
So cool.
This has been great, Amanda. We so appreciate this, and it sounds like you and DES and other groups in New Hampshire are doing a lot of work, but there's a lot more work to be done. And we can all help by keeping our eyes out, looking before we leap, and when we see something odd or unusual, take a picture and send it to you. So, any final words?
No, again, just please call me anytime, send me a photo. I would like to promote the EPA's Plume Watch app. It's not quite ready, but stay tuned for that, because I think we could get a lot out of also working with EPA on this project. So great. And in New Hampshire, in New Hampshire Lakes, we'd be happy to partner with you and DES and helping to get the word out even more. Because as you say, it's all about people being aware. So awesome. Well, thank you, Amanda. Thank you, Crystal and Jessica. Thank you to all of you for spending the last hour or so with us this evening. Again, in just a few minutes, you should get an evaluation in the email. Let us know how we did. Let us know what other things you want to learn. And tomorrow, again, you'll get links to the presentations. And if you haven't already, signed up for the next 3 series, because we are truly blessed to have experts like Amanda willing to spend their time and share their knowledge and ideas with you all. So thank you all. Be well. And if you're having a thunderstorm like I am, go enjoy it from a safe place. So, all right. Take care. Thank you. Thank you, guys.
Thanks, Amanda.